So that's all that says. So now to carry on from where we were before. Are there any questions so far? Okay. So we've got the nuclear capsid assembled, RNA nuclear capsid protein, and the polymerase. But we've also got a membrane component for this virus. It's an envelope virus. And so with the plasma membrane, what happens is it makes its glycoprotein, its G protein, by the standard way that the host cell makes transmembrane glycoproteins. So there's a signal sequence, it gets taken to the endoplasmic reticulum, the protein is extruded across the endoplasmic reticulum and is glycosylated in the luminal side. It's transported to the Golgi where more glycosylations take place, then buds off from the Golgi via vesicles which fuse with the plasma membrane, and in doing so insert the glycoprotein into the plasma membrane. So now you've got a plasma membrane which has got a glycoprotein inserted into it. Okay, so here's the plasma membrane with the glycoprotein inserted into it. And then what happens is the nuclear capsid comes and actually sort of pushes its way through the membrane, and in doing so gets coated with the membrane containing the glycoprotein. So now everything is assembling just nicely. And the nuclear capsid itself does not recognize the plasma membrane. What happens is there's this M protein, as in think of it as M for maturation, and that recognizes the cytoplasmic tail of the glycoprotein. And also it recognizes the nuclear capsid. And so it acts as the linking thing. So it grabs the glycoprotein with one hand, as it were, and the nuclear capsid with the other hand, and brings them together and enables them to package. And this is a feature you'll see of a lot of these envelope viruses. They're not all from the same family. Very different viruses all have got this principle of having a maturation protein. Okay, so now you've got new virus. So things to notice. The entire cycle here is in the cytoplasm. You can remove the nucleus and rhabdoviruses will still grow perfectly well. The RNA and the polymerase, the RNA polymerase and the modification enzymes, because those messages were capped and methylated and polyadenylated, all of that machinery is actually a part of this big polymerase. So that's coded for by the virus. Has to be. It's an RNA virus. And it's also present in the virion because the first thing it has to be done when it goes into a cell is to make message, so that all the machinery for making the message has to go in with it. And there's no early-late division. That's a common feature of these RNA viruses. They've got small genomes. They're pretty simple. They just do everything at once. Any questions? Okay. So now I'm going to switch to the paramyxoviruses, which are another non-segmented negative strand virus family. They're medically important, which is why we're covering them here as well. And they also introduce some additional concepts that I want to talk about, but in many ways their basic life cycle is very much like that of the rhabdoviruses. The members include parainfluenza virus, mumps, measles, and respiratory syncytial virus, and we'll be talking about all of those next week. Okay, so the rhabdovirus was a shrink-wrapped slinky, an enveloped helical nuclear capsid. This is also an enveloped helical nuclear capsid, but this is rabies, this is mumps, measles, parainfluenza. So we've got the very floppy kind of nuclear capsid in this. And also, this is shrink-wrapped in rabies, and this just has got a sort of big floppy bag on it in paramyxoviruses. So same basic structure, but they look rather different in the electron microscope. They are different families, they're not related. So you have this helical nuclear capsid, RNA, negative sense, non-segmented, with the polymerase associated with it. And then you have the envelope, which is shown in green. This has 2 glycoproteins inserted in it this time. Rabies had 1, paramyxoviruses have 2, and these are called the one type of spike is called either HN or H or G, and we'll go into why it's got different names in a minute, and the other one is called F. F is for fusion. And so this is the protein that's going to cause fusion. And then again, between the nuclear capsid and the membrane, there's this protein which lines the inside of the virus particle called M protein, and that too is involved in maturation just the same way as rabies that we just talked about. Oops. Oh, we don't need that. So why do we have these different glycoproteins? They're actually used as part of the classification of this family as to what kind of glycoproteins they have. So as I say, all members have 2 glycoproteins, all have an F protein, but some of them have these different attachment proteins, which is what this column is, the attachment proteins, H, N, H, or G. Now, the Paramyxovirus family has one subfamily called Paramyxovirus, or if you want to use the Latin, it's Paramyxoviridae, the family, and Paramyxovirinae is the subfamily. But most people don't use those terms, so I'm using what you will more frequently see. So the subfamily includes a genus called Paramyxovirus, a genus called Rubulavirus, and a genus called Mobilivirus. And Paramyxovirus genus itself include the human a couple of the human parainfluenza viruses we're going to talk about next week. Rubulavirus, it's got the same type of glycoproteins, but it's got some different features about its molecular strategy, and we're not going to go into those. But that causes these to be lumped together because they've got some things in common that are not shared by these members. And that's another couple of the human parainfluenza viruses and mumps, and morbillivirus are the measles virus, and also canine distemper in dogs. The pneumovirus family pneumo means air, lungs, so these are, like several of these are respiratory viruses. Its pneumovirus genus is respiratory syncytial virus, and the metapneumovirus genus, which is metapneumovirus. So we're going to be coming back to these again next week. But for the moment, what about this HNHG business? Okay, H stands for hemagglutination. And I know you've had hemagglutination in immunology, but I just want to refresh your memory because a lot of these viruses are hemagglutinating, and it's a very quick, easy test for viruses that have been widely used in clinical labs. So hemagglutination is something you see if the virus can bind to a red blood cell. Now, these viruses don't grow in the red blood cells. This is just dead end. But they happen to stick to red blood cells. And in fact, many of them don't stick to our red blood cells, but they stick to sheep red blood cells or goose red blood cells or duck red blood cells or chicken red blood cells. The lab pathologists, the lab people know which type of red blood cells they stick to. So if you stick to a red blood cell, here's a virus sticking to a red blood cell, probably not to scale. The virus probably should look a bit smaller than that, but anyway, it gives you some idea. So if it can stick to a red blood cell, that's because its attachment protein recognizes something on the surface of the red blood cell. So since the virus has got attachment protein all over its envelope, it can also stick, or you also get hemagglutinating viruses which are non-enveloped, but since they've got the attachment protein all over their surface, they can bind another red blood cell. And obviously this can bind more viruses, and then you can get a network where you've got all your red blood cells linked together in this network, which you see as a hemagglutination. So I just thought I'd show you, we used to have a practical many years ago, which has been dropped, but I was just going to show you how you look at these. Okay. You have these little plastic plates that you can set up your reactions. They're 96 well plates. And they also are numbered 1 to 12 and A through H down the sides. Okay, so you can put your red blood cells in here, along with some buffers so that they don't pop and all that. And then you can add the virus. And if the virus hemaggglutinates, it'll cause these networks to form. So if the networks form, you can look at these and see whether there are networks forming. Now, that's most easily done by looking up at the bottom, because what happens is this is not a proper hemagglutination plate. It should have pointed wells. So the red blood cells that don't hemagglutinate roll down into the bottom of the point, and you see a little pellet of red blood cells. It just occurs under normal. You don't have to centrifuge them or anything. It just occurs if you leave them for an hour or so at room temperature. The ones that agglutinate will form a network. So you could score your plate by doing this. However, some time ago, people came up with this high-tech version, which is if you do this, you can look in the mirror at the bottom and you can see the plate, and it's much more comfortable. Nowadays, there are machines that will read this for you, but this is a principle that's still widely used, and certainly outside in non-high-tech, it works perfectly well, because the reason hemagglutination is so useful is because blood is relatively cheap. You can bleed an animal as many times as you want to within reason. And the red blood cells are coloured, so you don't need any stains or dyes. And also, you're looking for a network. You're not looking for individual cells, so you don't need a microscope. So hemagglutination has been heavily used for viruses because it's been such an easy way. So what you do is you put the plate, and then you look down here, and here's my thumb. And if you look at the plate, what you can see is this is what the network looks like. So it looks like a sort of folded over skin, milk, like the skin you get on top of milk sometimes. It looks rather like that, folded over. So this is where there was enough virus to hemagglutinate. As they diluted out the virus, you can no longer see agglutination here, and you see these little pellets of red blood cells. So this is what a real assay looks like. And so you can see it's pretty easy to see when you no longer have enough virus to hemagglutinate, and this is the cutoff point. So you can read how many units there are here, and that tells you how much virus you have. If you have more viruses, it will go further out. If you had fewer viruses, it would come back here. If I took however much virus is present for, say, this cell, and added it to all the wells, I just took a complete line of red blood cells over here, and I added that amount of virus to every one of those wells, they would all agglutinate, and they'd all have agglutination in them. But then, if I took different concentrations of my serum and diluted it, if I got antibodies to the particular virus that was being used, then the antibodies would coat the outside of the virus, stop it binding to the red blood cells, and stop the hemagglutination. So that's hemagglutination inhibition. So if I've got the right antibody, I will stop the hemagglutination, and then you can see how far you can dilute my serum before the hemagglutination begins again, and that will give you some idea of how much antibody I've got to neutralize the virus. Neutralization means that the antibody will coat the virus or attach something on the virus in a way that stops it establishing an infection. But it's a fairish measure is to look at how much it stops hemagglutination. So hemagglutination inhibition is a useful way to see if I've been exposed to that particular virus. So if you use influenza A and I've just had flu, you can see whether my serum inhibits the influenza A hemagglutination or not. If it wasn't due to influenza A, it won't inhibit it. Any questions? Okay. So it's quick, easy, and cheap. You can use it to direct the virus or the antibody to the virus. And it tells you a measure of the total virus particles. In fact, you need an awful lot of virus particles to get a hemagglutination going, because you've got to have enough to cross-link all these red blood cells and make a visible network. But it doesn't measure infectious virus particles. It just measures total virus particles. So you can actually use it to measure the amount of virus in a vaccine, for example, even if it's an inactivated vaccine. It will tell you how many virus particles you've got, or at least a measure of them. But it doesn't actually tell you whether they're alive or dead. It's got nothing to do with that. And then there's another related aspect of viruses which hemagglutinate, and that is, if you grow your virus in tissue culture, it will put that glycoprotein, that attachment protein, into its cell surface. If it puts the attachment protein into a cell surface, then if you flood the culture with red blood cells, they'll bind to infected cells. And then if you rinse off the excess, you can see whether these will bind red blood cells. And infected cultures will bind red blood cells, non-infected won't. And you can see that here. And that's also widely used in the lab, because if you use heme absorption, again, it's very easy to see the red blood cells are brightly coloured for you. You don't need high-tech fluorescent microscopes or anything. And it also has the advantage that this is seen, you see the hemagglutinins in the membrane long before you start producing huge numbers of infectious virus particles. So it gives you a very early way to see if these cells are infected. So this happens to be with influenza, which is another hemagglutinating virus, but you can do it with any of the hemagglutinating viruses. Any questions on that? Okay. So the paramyxovirus subfamily all have hemagglutination activity. The pneumovirus don't. And because they don't have hemagglutination activity, it's known as G for glycoprotein, which is a terrible name for it because the point is the F protein, which is involved in fusion, is also a glycoprotein. But you have no control over why people give these names. So that explains why some of these have got the H. But why do these set up here, the parainfluenzas and mumps, why do they have N? And N stands for neuraminidase. And neuraminidase is an enzyme activity that destroys neuraminic acid. That's also known as sialic acid. And our cells are covered with sialic acid. So that is going to be important later on, and we'll come back to why that's so important later on. But neuraminidase is something that we will come back to. So that explains the N. And then the F, as I say, is because there's a fusion protein. And we've already discussed, when we talked about herpes virus, that fusion proteins that work at physiological pH will then cause infected cells to be able to fuse with each other, and you'll see giant cells and syncytial cells. And again, with a lot of paramyxoviruses, you see giant cells. This is just a review of that. So the virus binds to the cell membrane. The fusion protein, which in this case, unlike in rhabdoviruses, the glycoprotein and the fusion protein were part of the same protein. In this case, they're different proteins. And in this case, the fusion protein works at neutral pH, and it fuses directly with the cell membrane, and the nucleocapsid is delivered to the cytoplasm. And then it's the same story as we just went through with rhabdoviruses. When it goes into the cell, the transcriptase, the polymerase, is associated with it. It functions as a transcriptase. It makes individual messages, so that's how it gets around the monocystronic RNA problem, in a very similar mechanism, actually, to what rhabdoviruses do. And it's just got one more message, because it's got one more protein. It's got that extra glycoprotein. When there's enough nuclear protein made and it can start binding to the RNA as it's made, it then switches into the replication mode, and instead of making messages, it will now make a full-length copy that's coated with the nuclear protein as it's made. So again, this is the family jewels. It's protected by nuclear capsid as it's made, so it's always kept, and you don't get it naked. The naked stuff is the RNA. The family jewels are kept coated with protein. So for the negative strand viruses, it's much more like us. We keep our DNA largely coated with protein. The RNA it doesn't have all these protective proteins on it because we regard that as disposable. Same thing here, because it doesn't have to surrender negative strand RNA to the ribosomes. Glycoproteins, same thing, except that now we've got two glycoproteins and they're delivered to the surface via the Golgi. And assembly, same thing. You have the M protein functioning as a maturation protein, and the virus buds out. I didn't say at the beginning, it says on the slide, this is a pleomorphic virus because with this floppy nuclear capsid, floppy envelope, when you look at it in the electron microscope, you see very different shapes and forms because it just, particularly when it's dried out in the electron microscope, it assumes all sorts of appearances. It's not nearly as sort of highly structured as the rhabdoviruses are. But anyway, it buds out, and now you get the release virus particle. And now it should go off and live happily ever after, but there's a problem. And the problem is that I talked about these things bind to a receptor and get taken up into the cells. Well, in the case of paramyxoviruses, or at least in the case of these ones that we're talking about that have a neuraminidase, their problem is their receptor is sialic acid. So they bind to sialic acid. Now that's a problem because our cells, the surface of our cells is coated with sialic acid. We add sialic acid to lipids, we add them to proteins. Our glycoproteins have got a lot of terminal sialic acid on them. In addition to which, these are respiratory viruses that we're talking about. They come in through the respiratory tract. We have a lot of mucus in the respiratory tract, and that's rich in sialic acid. So this virus is, when it tries to leave the cell, it's got all these attachment proteins on the outside, and the cell itself, its glycoproteins, have got sialic acid on as well. So these will have sialic acid on, these will have sialic acid on. So if this virus gets out of the cell, it's coated with sialic acid. It's on all our glycoproteins. The cell is coated with sialic acid. And what's more, I don't have that. And if any other friends that have budded out at the same time will also be coated with sialic acid. So I've just tried to show that they're all coated with sialic acid. So this is going to mean that the attachment proteins that recognize sialic acid that have been inserted into the host cell membrane are going to bind to the virus particle as it leaves and try and pull it back because they'll bind to its sialic acid. Its attachment proteins will bind to the cell's sialic acid. So the two of them, it's like pulling Velcro apart. They're each going to want to bind to each other. So they're not going to be able to leave the cell. And in fact, in laboratory experiments, it's shown that if you have mutants that cannot get rid of the sialic acid, they actually have major problems establishing infections because they can't get away from the infected cell. So what the neuraminidase does, as I say, it chews up sialic acid. So it will remove all this sialic acid on the virus, and it will move. It's a part of one of these external proteins. It will remove all the sialic acid on the cell. So what you will have, instead of this sticking to this and not being able to get away, you remove or sticking to its fellows, you will remove all the sialic acid, and now this virus can float away. Of course, it will meet mucus as it floats away, and it will probably bind to the mucus, but again, this neuraminidase will digest the sialic acid off the mucus, and the virus won't get trapped. So this is really important, because otherwise the virus would just get trapped, and you just have a very local infection, and it wouldn't be able to cause all the nasty things that it does cause. So are there any questions about that? So it's basically a way to strip the glue off the cell. So if viruses bind to sialic acid, which is a receptor that's on so many cells, particularly respiratory cells, it gives them a lot of respiratory viruses do use sialic acid. If they have a receptor that binds to sialic acid, they're going to need a neuraminidase in order to get away. So the viruses that don't have the neuraminidase are not using sialic acid as their receptor. The other thing, while we're just talking about these properties of these proteins, in terms of getting out, is the fusion protein. So the F protein is the one that causes fusion at physiological pH. Now, if you think about this creates a problem because you're going to make this protein on your endoplasmic reticulum, or at least it's a problem for the virus. It's going to make its fusion protein, along with its attachment protein, on the endoplasmic reticulum. If this fusion protein starts causing fusion all over the place, then the endoplasmic reticulum is going to be fusing, and the different layers of the Golgi are going to be fusing, and this nice directed ability to move from endoplasmic reticulum to Golgi apparatus to Golgi vesicles that go to the cell surface is all going to be disrupted because you're going to have membranes fusing everywhere, and it's going to be a mess. So the fusion protein can work at acid pH, but when it's made at a neutral pH, when it's made, it doesn't work. And I just wanted to go over this because this is actually also a target for chemotherapy. Currently in trials, but not yet on the market. So the area that causes fusion between two membranes at physiological pH is actually not at the end of the glycoprotein, it's actually a region in the middle. And this just shows that there's a disulfide bond here that bridges this area. What happens is this is not active in fusion because the end that's going to sort of bury itself in the membrane and facilitate fusion is tied up with the rest of this protein and it can't get away and work. So it's like, it's this hand is going to grab the membrane and cause fusion. But if it's being held by something else, you can't use the hand. So how's it going to work? And the answer is you chop the something else off, and now this hand is free and it can go and cause fusion. And so that's what happens here. There's a protease that cleaves this and enables it now to flip out and cause fusion. That protease is one of our proteases, because we also have proteins that we don't want to be activated until the last minute. And so we also have proteases that cleave proteins, but only just as they're about to leave the cell, because we don't want to be active in the cell that they were made in. So it uses the same system. So this is a host cell thing. So as it goes through here, inactive, as it goes through here, inactive, inactive, as it goes through here, inactive, and as it's getting towards the surface, these vesicles contain a protease which will cleave it. And so by the time it gets to the surface, it's now active. And as I say, those proteases cleave some of our proteins, again, so that they're only active as they reach the cell surface. And this is a host cell. So if we could do without it, it would be a good target. But okay, so any questions? So as I say, the basic part of the life cycle is very similar to rhabdoviruses. But the difference is, there's a difference in shape. They're both helical RNA enveloped viruses, but the rhabdoviruses have a bullet or bacilliform shape. These are round and tend to be a bit pleomorphic. Rhabdoviruses have one glycoprotein, which is the attachment protein, and once it gets into acid vesicles, it's also a fusion protein. These have two. One functions in attachment, one functions in fusion. And the fusion pH is here for the rhabdovirus is acidic, so it has to be taken up into the cell. Here it's neutral, so it just fuses directly with the plasma membrane. Okay.
The F protein is inserted in the infected cell membrane. Correct?
The F protein is made just like the attachment protein. They're both glycoproteins. The message for both of them codes for a signal sequence which will take both of them to the endoplasmic reticulum, and then they're made just like our regular glycoproteins. So they're made as transmembrane glycoproteins in the endoplasmic reticulum. They get more processing of the glycosylated portion in the Golgi. Then they are pinched off from the Golgi in these Golgi vesicles. The vesicles go to the plasma membrane, fuse with the plasma membrane, and deliver these transmembrane proteins to the plasma membrane. It's the same way that you get your MHC glycoproteins there and all our other glycoproteins.
And the fusion proteins are what facilitate more virus attachment to the infected?
No, the fusion. What happens when the virus leaves the cell, and leaving the cell is helped by this neuraminidase activity that enables it to get away. And the fusion protein has now been activated because it was activated at the last step as the virus got out of the cell. So when it meets the new cell, it's got a hemagglutinin and a fusion protein. The hemagglutinin binds the virus to the cell, and then the fusion protein causes the fusion. So they're just, in the rhabdoviruses, they both do it together. In this one, there are two separate proteins. Okay. Any other comments? Okay, so now I want to change gears and talk about still negative strand RNA viruses, but now we're going to come to segmented negative strands RNA viruses, where the genome is distributed between different bits of RNA. And the ones we're going to talk about, the only one I'm going to talk about in this set of lectures, are the orthomyxoviruses. And these are the true influenza viruses. So para-influenza viruses are not the true influenza viruses. They tend to cause, they're different, very different. So these are the true influenza viruses, which are the classic influenza. So similar thing, helical RNA nucleocapsids, rather more the telephone cord model. But this time, there are, in the case of influenza A and influenza B, 8 segments here. And again, the polymerase complex is associated with those. So when it's made, it has an affinity for nucleocapsids and binds to them. Again, it has an envelope. And again, that functions in maturation, similar to the story that we've already had for the other two. And again, there are two glycoproteins in that envelope, just like we saw with paramyxoviruses. This time, there's a different distribution of labour. There's a hemagglutinin, and there's a neuraminidase. So the neuraminidase is on a separate protein here, and the fusion activity is actually on the hemagglutinin. So different division of labour between flu and orthomyxoviruses and paramyxoviruses. We'll go into those in a bit more detail later on. So I think, again, they're a bit pleomorphic. When you look at them, you can see they adopt rather different shapes in the electron microscope. In fact, these were ones grown in culture. When they're grown in culture, they tend to look much more alike. When you actually isolate them from respiratory secretions, they tend to look much messier. So this is a swine flu, actually, which is isolated from respiratory secretions. And you can see that it looks a bit of a mess. Okay, the absorption penetration thing is what should be now really familiar. It binds to receptors on the cell surface. In this case, they include sialic acid. So this virus is going to need a neuraminidase. So the HA binds to the sialic acid. Taken up into vesicles, these do not fuse directly with the cell. We said only herpes, paramyxo, and HIV do that, of the ones we're going to be talking about in either part of the virology course. So here, they get inside, the nuclear capsids are released because there's fusion with the endosome membrane. And then they go to the nuclear pore and they go into the nucleus. So this is the first of the RNA viruses that we've talked about that goes to the nucleus. We said there's not too much in the nucleus. We can't replicate RNA, we replicate DNA. So they can't get our replication machinery. We can't copy it into messages because our RNA polymerases use DNA as a template, and the viruses would use RNA as a template, and we don't, our polymerase won't. We can't use them to cap and methylate and polyadenylate our messages, because in our cells, polymerase II goes around as a complex with all the modification machinery, and it will only modify the RNA that it's just made. So the RNA has to be modified before it's released. So it goes through this complex, and it doesn't grab an RNA from elsewhere in the nucleus and cap and methylate it and polyadenylate it. POL2 does it in association with its friends, and they go round as a gang. So what's it going to the nucleus for? And actually, about the only thing that you can see that's really available in the nucleus that's not available in the cytoplasm is splicing. So you expect these viruses that go to the nucleus to have splicing. Okay. So this virus goes to the nucleus, and of course there are 8 segments, but we're just going to focus on looking at one. The others all function the same. So what does it do? Well, it's got the problem of how is it going to cap and methylate its RNA? And it has a solution that other viruses have come to, none of the ones that we've talked to so far, but there are viruses that grow in the cytoplasm that have also come to the same solution. One easy way is not to make an enzyme that does this. One easy way is to actually do piracy. So what they do is they take host cell messages and they cleave off a bit at the 5' end. And you don't need to know this, but it's about 20 nucleotides. And they cleave it off. And then that, of course, has got a methylated cap. And then they use that as a primer for RNA synthesis. And I've shown here that it ends with an A just because it will make more sense to you in the next stage. You don't need to remember that. But if, say, this ends in an A, the first base on the influenza negative strand nuclear capsid, and negative is shown in blue, is a U. So this A will pair with this U, and then the polymerase will extend it, which is the viral polymerase, not our polymerase, because, as I say, it's an RNA template. So the viral polymerase, which had to come in with the virus particle, of course, along with the endonuclease, which also had to come in with the virus particle, the polymerase will now extend this message and make a viral messenger RNA. But every viral messenger RNA will have a bit of a host cell message at the 5' end. So this was originally called piracy by the people who originally discovered it, and it's a nice term. So they go around snaffling caps from messages. One thing is, once you remove a cap from the message, it no longer has a cap methylated 5' end, and so the messages it takes the caps from will actually be rapidly degraded. So our messages start being degraded as a result of having lost their caps. So the RNA synthesis needs the endonuclease, which is virally coded and is taken in the virus particle because it has to function in order to make the first messages. And there's a polymerase which is also virally coded and taken in with the virus particle. And a poly-A polymerase which is also virally coded and taken in with the virus particle. And I've already said why doesn't it use host cell enzymes? Because our enzymes aren't available. So but as I say, it will use splicing. And so several segments. It looks like a good message now. It's got a 5' capped methylated end. It's got a 3' polyadenylated end. It looks like a good message. And several of the messages do have the sequences for intron removal. And so they can code for several proteins, because if you don't remove the intron, you get this protein. And if you do remove the intron, you get a different protein. And we will come back to that when we talk about flu. But several segments have got this. So by going to the nucleus, it has the option to use splicing to get more than one message out of one of these genome segments. All the others just give rise to one message per segment. So it makes the messages go to the cytoplasm, the proteins are translated, they go back into the nucleus, and when there's enough of the nucleocapsid protein, it's the same story as we saw with the rhabdoviruses and the paramyxoviruses. Once there's enough nuclear protein, it switches over to, instead of making messages, it makes genuine copies of the genome. So there's none of this 5' end coming from the host cell messages anymore. It's now an accurate copy of the genome from one end to the other, because this is replication. And as it replicates it, it's coated with that nucleocapsid protein. So again, it keeps the family jewels protected by nucleic acid, by nuclear protein all the time. And again, there's no clear early late. It just all goes off in one go. These are simple not simple, but small viruses in terms of genome. So you make the nucleocapsids in the nucleus. We can get nucleocapsids out of the nucleus. After all, we get ribosomes out of the nucleus, so nucleocapsids are not apparently a problem. So these helical nucleocapsids can be transported out of the nucleus into the cytoplasm. Meanwhile, the glycoproteins were made on the endoplasmic reticulum, went to the Golgi, went far vesicles to the plasma membrane. The vesicles fused with the plasma membrane and delivered the glycoproteins to the plasma membrane. So we got the hemagglutinin and the neuraminidase in the plasma membrane. Again, there are maturation proteins which act to grab the inside tail of the glycoproteins and the nucleocapsid and bring everything together. And they bud through the plasma membrane. And we've got no idea how they ensure that they get one copy of each genome segment. We suspect that what they do is package a few extra segments so that they hope that they've got one copy of everything. And probably that explains a lot of the reason why not all virus particles are infectious. They don't all have a complete copy of the genome. And we need to talk about cleavage for the fusion protein, as we did before, and role of the neuraminidase again. Okay, so the hemagglutinin binds to sialic acid, and it's also the protein that causes fusion once it gets into acid endosomes. So what happens? What happens is it has to have two steps. So I just wanted, this is my old cell phone, and it has a rubber band around it. What it needs to do is, once it gets outside the cell, this is not cleaved inside the cell, it's cleaved predominantly in our extracellular fluids. We have proteases in our extracellular fluids. We think they're protective, but flu thinks they're wonderful. So what happens is, this glycoprotein, it can bind at the moment to sialic acid, but it can't cause fusion. And the only way it can cause fusion is if you clip something so that it's now free to go but it still stays in the closed conformation. So nothing will happen, but at least you've removed one object. And then when it gets inside the cell, inside the next cell that it's going into, it will be taken up into endosomes, which will then become acid. Under the acid pH, it undergoes a conformational change, flips over, and now this surface can cause fusion. So what you have to have is two steps for flu. One is a protease in our extracellular fluids to clip that band that was stopping it open anyway. And two is you actually have to flip it open by the conformational change that occurs inside the endosome. So it needs both things in order to get going, which stops it doing inappropriate fusions. So there are some drugs currently under study, and they're at least into early trials, which are actually protease inhibitors, with the idea that they inhibit that protease that you've got in your lung, in your respiratory fluids, and stop this activation of flu. Any questions on it? Okay. So that just shows you what happens. I've gone through that. And then this is just a table that summarizes the differences between the paramyxoviruses and the flu viruses, segmented versus non-segmented genomes, and that has a big influence on the epidemiology. Cytoplasmic versus nuclear, different primers. Hemagglutinin and neuraminidase are on different proteins, and the fusion proteins function at different pHs. And I will talk about retroviruses on Wednesday. So thank you, and good luck in the exam.